Thanks so much for having me today. It's great to see a lot of familiar faces. Hard to believe that I've been at Regeneron now for 4 years, and also, as Rochelle said, I've also maintained a part-time appointment at the University of Maryland. So I'm back and forth a fair bit between New York and Baltimore. And my task today is to tell you a little bit about what we've been up to at Regeneron with regard to now how we're applying high-throughput human genomics for novel gene discovery. In a very blue-sky way, we're interested in understanding human biology, and in so doing, ultimately identifying new drug targets. So that's essentially what we're up to, and the motivation behind this and the reason why the pharma and biotech industry has a renewed interest in human genetics is shown on this slide, and that is that the way we do business, have done business in the past, has been incredibly inefficient. A lot of R&D being done, billions of dollars, in fact, in the pharma industry, and very few, relatively speaking, new molecular entities have been hitting FDA approval and ultimately patients. And this is because more than 90% of molecules that enter into humans, into phase 1 clinical trials, don't demonstrate sufficient safety or efficacy to ultimately make it to the finish line. And most of these failures occur in phase 2 clinical trials where hundreds of millions, if not more, dollars have been spent in the development of that particular molecule. And this is due largely to lack of efficacy and also, in many instances, toxicity and the combinations of efficacy and toxicity thereof. And this has led, essentially, the field to come to the conclusion that preclinical models are really poor predictors of clinical benefits in man. And now several examples with high-throughput genomic approaches that have shown that compounds that are supported by human genetic evidence are more likely to succeed in terms of being effective and safe for humans. 2 good examples are shown on this slide. Well, actually, I see it, but I can't advance. Is there a way to advance the slides? Maybe if I go to here. I don't know, it's... Oh, maybe the remote, okay, let's try that. Okay, there we go. All right, so 2 good examples of how human genetics has informed drug development are shown on this slide. The one on the top, well-known PCSK9, where families had been identified back about 2003 that had gain-of-function variants in this gene that caused a new form of familial hypercholesterolemia. Population studies that showed not there long after that loss-of-function variants in this same gene led to people that had very low levels of LDL cholesterol and were protected from coronary heart disease. This identified PCSK9, specifically inhibition, as a terrific target potentially for LDL lowering, and ultimately clinical proof of concept and ultimately FDA approval of now 2 monoclonal antibodies that inhibit PCSK9 now available for treatment of hypercholesterolemia. The lower one that I'm showing you I'm particularly proud of because the initial finding was discovered in the old order Amish. Actually, my group back in 2008 identified for the first time humans with null mutations in ApoC3. These individuals had very low triglyceride levels. They had less coronary artery calcification, and they actually lived longer. And this led to population studies that showed that these variants occur in the general population and indeed certainly do protect from coronary heart disease. And now in clinical trials, antisense inhibitors of ApoC3 that are in phase 3 clinical trials for the treatment of hypertriglyceridemia and coronary artery disease. So this has led to the idea that with high-throughput human genomics, we can probably find many of these examples, these genetic experiments of nature that can inform human biology and ultimately do new therapeutic targets. And the Regeneron Genetics Center, as well as other pharma and biotech industry groups, have been working very hard now in the human genomics arena. Very interested, as I've mentioned, in the area of target discovery to identify new drug targets. As I'll show you, you can use human genetics to identify indications for a particular drug target. One can use genetics, human genetics, to de-risk a particular drug target. For example, to confirm lack of on-target adverse side effects in the drug target in individuals who carry loss-of-function variants in that drug target. And, of course, very interested in more traditional pharmacogenetics, biomarkers that would mark individuals who would be more likely to be responders or non-responders or have adverse effects from a drug or not. I'm not going to say too much more about the biomarker arena of pharmacogenetics, except to say that at Regeneron, as a policy, we collect DNA samples on everyone from all of our clinical trials to the extent that informed consents and local IRBs will permit, and we perform full exome sequencing and genome-wide SNP chips for pharmacogenetics in all of our trials from phase 1 all the way up through phase 4 clinical trials. So I'm going to concentrate today more on the target discovery and indication discovery side of things, since there seems to be a lot of new and interesting advances in that arena. The Regeneron Genetics Center now is about 5 years old, and we work with academic collaborators around the world to gain access to DNA samples from well-phenotyped individuals from around the world. We now have over 60 academic collaborations, and I just heard a couple of days ago, we're now well over 400,000 exomes that have been sequenced at the Regeneron Genetics Center in Tarrytown, New York. You can see that we're very interested in multiple study designs. We're very interested in general populations, particularly those that are linked to de-identified electronic health records, such as the Geisinger Health System, UK Biobank, and the like. We're very interested in large case control cohorts for phenotype-specific analyses. Very interested still in the undiagnosed Mendelian space, family studies of probands and family members that have rare undiagnosed Mendelian-like diseases. Can certainly inform human biology. And of course, near and dear to my heart, and I'll come back to this in the latter part of my talk, the founder and special populations that provide unique opportunities for novel gene discovery. So I'll say something about the Regeneron Genetics Center and our Geisinger collaboration, the so-called Discover collaboration. Geisinger Health System, as many of you know, is an integrated healthcare system in central Pennsylvania. They care for about 1.6 million participants, predominantly of European Caucasian ancestry. They were early adopters of the electronic health record and have longitudinal healthcare record for a median of about 13.4 years. And now, several years ago, the Geisinger Health System began their MyCode biorepository. They've now recruited well over 150,000 patients into their biorepository, and through a collaboration with Regeneron, we've now exome sequenced and genome-wide SNP chipped over 90,000 of these individuals. These are patients that have been recruited largely from an unselected population within the Geisinger Health System, and of course we've also been able to focus in specific areas like cardiac catheterization lab, bariatric surgery, and some other places. So I'm going to summarize just in a couple of slides what we've learned about exome sequencing of the first 50,000 patients from the Geisinger Health System. I should say that all of this sequence data, the variants, are publicly available data on the website that you can see in the lower right-hand part of the slide. So if you're interested in knowing whether a variant was detected in these 50,000 exomes, you can go right to discovershare.com. This slide quickly shows you the very rich longitudinal electronic health record data that we have as part of the Geisinger Discover program. You can see on top that there are many years of data in most Geisinger patients. They tend not to move, and they're happy with their healthcare system. About 400,000 patients have at least 10 years or more of electronic health record data in the Geisinger database. You can see there are millions of laboratory tests from clinical chemistries and hematology on the lower left-hand part of the slide. And if you're looking at diagnoses of common and even not so common diseases, you can see hundreds of thousands, tens of thousands to hundreds of thousands of cases that one can derive from the Geisinger electronic health record. So that's the clinical phenotype database. In terms of the genetics now, having sequenced 50,000 exomes, we can summarize what the variation looks like in this relatively unselected patient population. Again, these are exomes, not whole genomes, and you can see that we've identified a bit over 4 million variants in these 50,000 or so exomes. You can see that the vast majority of these variants are very rare. They have a frequency of 1% or less. We've identified about 176,000 variants that would be predicted to be loss of function variants in these genes. And if you look at a given individual, we can tell you that on average, each individual is heterozygous for a predicted loss of function variant for about 21 genes, and each individual is homozygous for a predicted loss of function variant in approximately 1 gene. And finally, a big question occurs as to what does this mean for an integrated healthcare system in terms of return of results. And in our initial calculations, we found that if you look at the ACMG 59 and then some additional genes that Geisinger has determined are actionable results, about 3.5% of these 50,000 individuals that were sequenced in this integrated healthcare system have an actionable result that would warrant return of results through genomic medicine clinics, genetic counseling, and the like. So this is kind of what we need to be equipping our healthcare system for as whole exome sequencing becomes more the standard of care. So the other thing we can do now with 50,000 exomes is actually do this proof of principle experiment where we can ask the question, would we be able to have discovered drug targets that we already know are effective, that have been FDA approved and are already effective and safe? And so we performed this proof of principle experiment where essentially we looked at all of the targets for lipid-lowering therapy, those that were FDA approved and those that are in phase 2 and 3 clinical trials that appear that they will be effective, safe, and ultimately FDA approvable. And we asked, could we find loss of function carriers in these genes that would have predicted that therapeutic effect? And the bottom line is that we were able to detect in 8 out of the 11 lipid therapy targets individuals with loss of function variants in which these individuals had nominally significant or directionally consistent clinical associations that recapitulates that drug effects. And I'll show you just a few examples. ApoB loss of function carriers have on average LDL cholesterol levels that are 15 milligrams per deciliter lower, suggesting that ApoB antagonists would be effective lipid-lowering agents, and we already know that that's true. PCSK9, already you've seen some of that data. Again, genetics would have predicted that PCSK9 would be an effective drug target. My friend ApoC3, same thing, loss of function carriers have much lower triglyceride levels, would have predicted in advance, or would have predicted based on genetics that this would be a good target. And in a slide or two to follow, I'll tell you a little bit about the bottom row, angiopoietin-like 3. This would predict inhibition to be effective triglyceride lowering. And one question in addition was whether potentially, in addition to triglyceride lowering, this might also be a cardioprotective therapeutic. And now in 2 publications, we've been able to show that both angiopoietin-like 3 loss of function carriers as well as angiopoietin-like 4 loss of function carriers, not only do they have lower triglyceride levels, but they also appear to be protected from coronary artery disease. Again, proof of principle suggesting that this would be a good drug target for triglyceride lowering and also cardioprotection. And more recently, we've been able to find potentially a new indication for angiopoietin-like 4 inhibitors, in that these individuals that carry these loss-of-function variants in angiopoietin-like 4 also appear to be protected from type 2 diabetes. We would not have ordinarily predicted that based upon what we knew or thought we knew about the human biology, but here we have genetic proof of principle that an indication here, in addition, could potentially be for type 2 diabetes. So I'd like to tell you the story now about a completely novel gene that we came across that potentially could be a great drug target for chronic liver disease. Many of you know that non-alcoholic fatty liver disease is very common in our obesogenic environment, and now one of the major causes of liver failure, also hepatocellular carcinoma. And this story starts with an exome-wide association study of liver function tests in about 46,000 patients from the Geisinger health system. And you can see that we picked up many of the already known genome-wide significant hits for ALT levels and AST levels. And you can see with the blue arrow on the left, a novel hit, a SNP in a gene called HSD17B13. When we look further at that variant, you can see on the right, to see whether that variant was associated with liver pathology. You can see in the Geisinger data set, sort of no matter how we slice the liver pathology, this variant appears to be protective from that liver pathology, whether we looked at alcoholic liver disease, non-alcoholic liver disease, cirrhosis, even hepatocellular carcinoma. And those data were nicely replicated in an independent study in collaboration with the Dallas Liver Study. You can see on the bottom right, protection of individuals with this variant from chronic liver disease. We also had access to liver biopsy samples from about 2,000 Geisinger patients who were part of their bariatric surgery program. And in those individuals, this particular genotype was associated with pathological hepatosteatosis. So you can see from this slide that there was not association with simple steatosis and this variant, but with the more pathological signs of steatosis, that of non-alcoholic steatohepatitis, NASH, NASH with fibrosis, and also cirrhosis. So this is a variant that we believe is not protecting from fatty liver, it's protecting from the progression of fatty liver to more pathological forms of fatty liver disease, namely hepatitis and cirrhosis. It turns out that this particular variant is a TA insertion, and it occurs at a splice site in one of the exons. And actually, when we looked at mRNA splicing from human liver samples, we were able to show that this TA variant actually alters a splice site and results in an altered mRNA splice. You can see that wild-type TTs on the upper right produce almost exclusively the A transcript, the mature mRNA that encodes the mature protein. And you can see those who are TAs and TA homozygotes specifically make hardly any of the mature transcript, but more of the alternate transcript, what we call transcript D. This is an mRNA that results in a frame shift and an early chain termination, and likely, as I'll show you in the slides to come, a protein that is inactive. Protein in these variant carriers, you can see in slide B and insets B and C that the protein, the mature protein, is not produced in these TA homozygotes. And in histochemistry, you can see that this is a protein that really very little is known about in terms of its function, but we now know that it localizes to the lipid droplet in HepG2 cells. So HSD17B13 is a member of a large family of enzymes, the hydroxysteroid 17-beta dehydrogenase family. And this is a family of proteins that's involved in metabolism of sex steroids and also fatty acids. The endogenous substrate for this particular enzyme is not known. This led to in-house testing of hundreds of putative substrates in an in vitro assay, and we were able to identify estradiol as well as some of the bioactive lipids such as leukotriene B4 as substrates for this enzyme, as you can see on the upper right-hand slide. And we were also able to show that the isoform A was an active enzyme, whereas the isoform D, encoded by the alternate splice, is essentially an inactive enzyme, suggesting here that inactivity of this particular enzyme protects from chronic liver disease. So that's a cool story about large exome-wide association study that led to a new drug target, potentially for a disease of major clinical importance. And efforts continue to find small molecules that will inhibit this particular enzyme as a potential therapeutic agent for chronic liver disease. And so I'll move now to a second story that I think is pretty cool. If any of you thought that you'd get away with not hearing about founder populations and the Amish in any talk of mine, fear not. Founder populations, I still believe, are really important for novel gene discovery, and some of the principles of population genetics is shown on this slide. Founder populations have marked genetic homogeneity because, of course, by definition, the gene pool of the entire population derives from a small number of founders. And then through the generations, genetic drift can result in a rare, even a single copy of that founder allele to be amplified to be markedly increased in frequency. And this is what provides that genetic experiment an opportunity in which that variant can now be identified and ultimately function can be defined. So we're particularly interested in large effect, loss of function variants that have gotten into the population and that have drifted up in frequency and therefore could have only been discovered in that population. And in addition, founder populations, many of them practice consanguinity, or endogamy, I should say, and therefore consanguinity can then lead to homozygosity for these drifted alleles and ultimately recessive effects. Finally, founder populations tend to have a much more homogeneous lifestyle, and that we believe will introduce fewer confounding influences to identify larger genetic effects. So this is why we're quite interested in founder populations at the Regeneron Genetics Center. And one thing that I established shortly after arrival is the DRIFT program for discovery, research, investigating founder population traits. You can see that we're interested in hundreds, even thousands of founder populations from throughout the world, since each is its own experiment of nature. We're interested initially in just cataloging the population-specific allelic architecture. We also then want to understand the biological function of drifted alleles, both loss of function and gain of function mutations in these populations. And for many of these populations, we can also engage them in callback studies where we can bring individuals back of specific genotypes of interest so that we can dig deeper into the human biology and physiology. Currently, the DRIFT program has 15 academic collaborators from around the world, and they have provided access to over 180,000 samples from more than 80 distinct populations, founder populations from around the world. And you can see some of them here on the map. So a really rich resource for genetic discovery. This slide just shows you a summary of kind of what we're beginning to find about with regard to the allelic architecture across distinct populations around the world. And this is sort of a little bit of comparing apples and oranges because the sample sizes are different among these populations, but I think it makes the point pretty well that if you look at the Geisinger European ancestry group, 60,000 exome sequence, that's sort of a general European Caucasian population, and you compare them to, say, the Pennsylvania Amish or the Ohio Amish, Southwest Native Americans, Ashkenazi Jewish, Pakistani. You can see that each of these populations have loss of function variants that they share, but each also has its own unique loss of function variants, really suggesting that sampling many distinct founder populations from around the world are opportunities to capture impact of variation of genes at appreciable frequencies that you really can't find in even large numbers of individuals from general populations. And of course, endogamy here, especially in the Pakistani group, helps quite a lot in terms of identifying homozygotes for loss of function variants. So this is sort of a proof of principle showing that founder populations from around the world, we're not going to exhaust the number of the loss of function variants that we find. Each population will have its own cadre of loss of function variants that are amenable to study. Okay, so one founder population that we've now made the most progress with regard to novel gene discovery and potential new drug targets is the old order Amish. And this is a homogeneous founder population that's been studied for a very long time, from the time of Victor McCusick and his studies back in the 1950s. And over the years now, the University of Maryland group has now recruited over 7,000 Amish individuals from Lancaster County, PA, that we have now exome sequenced at the Regeneron Genetics Center. And this has been really satisfying to me since over 25 years at the University of Maryland. I actually collected most of these samples at the Amish Research Clinic. We've made some cool discoveries along the way, but now we feel with full exome sequence and genome-wide SNP data that for the first time we're literally finally playing with a full deck and are really discovering some very cool things. So this is a population founded by 200 to 600 individuals who migrated from Switzerland about 15 generations ago. The current consensus size is around 40,000 individuals. They've remained an isolated, homogeneous founder population because of their cultural and religious traditions. There is endogamy. Average relatedness is first cousins once removed. And they have very large SIB-ships, which actually facilitates recruitment of individuals and individuals that have rare alleles that can segregate in families. As I've mentioned, over the years, we've made a lot of cool discoveries just with small amounts of genetic data. This slide kind of summarizes what we know, at least up until the time of exome sequencing, and then I'll tell you some two new stories with regard to exome sequencing. We know that 1 out of 25 Amish carry that null mutation in ApoC3. These individuals, as I mentioned, have low triglyceride levels and are protected from coronary artery disease. Many of you know through the work that we've done with the Pharmacogenomics Research Network, the first genome-wide association study of clopidogrel responsiveness was done in the Amish, the finding of CYP2C19 as a major determinant of clopidogrel response. It turns out that 12%, 1 out of every 8 Amish individuals, has familial hypercholesterolemia due to a mutation in their ApoB gene, and these individuals have LDL cholesterol levels that are on average 60 milligrams per deciliter higher per allele than those without this variant, so a major cause of hypercholesterolemia in the Amish. A very cool null mutation in LIPE, hormone-sensitive lipase. These individuals have a higher risk of type 2 diabetes, and the 4 homozygotes that we identified have lipoatrophic diabetes. And through exome sequencing, not only with my colleagues at the University of Maryland, but also through the Clinic for Special Children up in Lancaster, PA, we've identified now dozens of new genes for monogenic diseases in the Amish and the Mennonite community, those that are now being used actually for clinical care in the population. So I'm going to tell you one news story in the time remaining, and this is a cool example of a novel gene that can only be discovered in a founder population like the Amish that potentially introduces opportunities as a new drug target. Some of this data will be presented in much more detail at the ASHG meetings to come in the next few days, and the abstract numbers are shown here on the slide. So this is what an exome-wide association analysis for LDL cholesterol looks like in now about, this was about 5,000 Amish individuals. You can see that ApoB hit that I told you about before, very strong association with LDL cholesterol levels. But what's new here is a new hit on chromosome 9, an exome-wide hit on chromosome 9, completely novel. And as it turns out, this is a variant that's not present in any of the huge genome-wide association studies of lipids. And the reason is that this is a drifted allele that has a frequency of about 6% in the Amish. You can see in the Geisinger data set, very, very rare. In NOMAD, only 2 out of 277,000 individuals carry this particular variant. So this is a highly drifted allele and is in the Amish highly associated with lower LDL cholesterol levels. And that's what this slide shows, p-values of 10 to the minus 9th and 10 to the minus 10th of associations of this particular variant. It encodes a missense variant, asparagine 352 serine, in a gene called B4GALT1. We're also interested in this variant because there's also nominal association of this variant with lower fibrinogen levels. You can see on the lower part of the slide. And this is, fibrinogen is actually an independent risk factor for coronary artery disease. So the idea here being that potentially this could be a drug target that protects from heart disease both by lipid lowering as well as potentially acting on the coagulation cascade by decreasing fibrinogen levels. You can see also nominal association for lower coronary artery calcification, again suggesting a cardioprotective effect. So what is B4GALT1? It turns out that this is a galactosyl transferase, and it's involved in the ER and the Golgi to essentially build glycan chains on glycoproteins. Specifically, it will add a galactose to the growing chain of these glycoproteins. And as it turns out, this particular variant, asparagine 352 serine, is very close to the active site of this enzyme. So we were able to do a freezer experiment where essentially we got plasma and serum out of the freezer from these patients that have, or I should say subjects that are homozygous or not for the serine residue. And we were then able to do, using LC-MS, end-linked glycan analysis, to actually see whether glycoproteins were different in these individuals or not. And to make a long story short, they certainly are. You can see on the top-hand slide global glycans in all of the plasma. On average, individuals that are homozygous for the serine allele have less galactose on their glycoproteins, also less sialic acid. When we start to do immunoprecipitation for specific proteins, you can see we have a marked decrease in galactose and sialic acid on fibrinogen and as well immunoglobulin. And we're now digging more into the specifics of what glycoproteins are relevant here for the phenotypes. We're showing you here an example of ApoB, where we also have an example that there's much less galactose and sialic acid on ApoB, which we believe might be one of the reasons for the lower LDL. We have knocked down B4GALT1 both in zebrafish and in mice. This is the data in zebrafish, where you can see the morpholino knockdown results in a decrease in LDL levels in the zebrafish that can be rescued with the wild type B4GALT1 and only partially rescued with the mutant B4GALT1. And finally here, just to show you, we can also knock down B4GALT1 in mouse liver in vivo, and in red you can see about a 50% reduction in LDL cholesterol in the knockdown mice. So we also know that B4GALT1 may have additional roles in immune function, cancer, lactation, and reproduction, and this is why we're quite interested in understanding in a deeper way the biology of this galactosyl transferase. So just to summarize and conclude, I believe the future of drug discovery is really bright because it's going to be fueled by human genomics, and we're now at a state where we can sequence hundreds of thousands, even millions of individuals to make these genetic discoveries. Genetic experiments of nature, whether it be with large patient populations or founder populations, are going to be the targets for which we will exploit human genetics for novel target discovery. I've shown you some examples there. And in addition, maybe through examples here, I've shown you how partnerships between industry, academia, and healthcare systems will fuel this new age in genomic discovery and also the implementation of precision medicine in our healthcare system. So with that, I think I'll conclude. Huge numbers of individuals at Regeneron as well as collaborators at Geisinger and University of Maryland and some of our other institutions as well. Thanks for your attention. Happy to answer questions. Or questions later. Now? Okay.
Are you pursuing the idea of a drug to prevent diabetes? How would you develop that?
A drug to prevent diabetes, and how would one develop that? So, I have to say, a lot of pharma and biotech companies have started to move away from type 2 diabetes because there are already some pretty good medications, and also the FDA has kind of mandated these outcome studies that are quite difficult and expensive to do. Our CSO, George Yankopoulos, tells us that focus is a dirty word. He says that every day. And that we should be doing blue sky science and blue sky human genetics, identify new biology, and then we figure out where it goes. And so type 2 diabetes is on the table and of great interest to us. Now, in terms of how would you design a trial for prevention, it's going to require a long-term trial. There's just no question about that. But I think if you choose the patient populations appropriately, in terms of those that are at marked risk of developing diabetes, say within a 5-year period, you might be able to be within a window of time that a clinical trial is reasonable to conduct.
Genetic mutation may be different from than adding the drug after development. So in some cases they work, in others not. Is there any thoughts you have?
Right. So the point Wolfgang is making is that genetics is sort of one piece to the puzzle, and these loss of function variants in humans can certainly inform human biology, but they might not all translate to actual effective drugs. And I think that point is well taken. I mean, we see the positives in applying human genomics for novel gene discovery, but there are a number of factors that are in play that may make genetics less of an attractive alternative. One is, of course, that these variants are present from birth, whereas a therapeutic you're going to be adding on presumably in adulthood when a disease manifests. So that's certainly a difference in the paradigm. So I think the word is out in terms of what proportion of drug targets discovered through genetics will ultimately be successful. But I could tell you, without human genetics, we're starting with a pretty low bar, and we can only believe that genetics will contribute in a positive way.
So if you think about genetic drift, I think a lot of times the variants with a modest effect on fitness will be enriched because of genetic drift. But things with severe damaging function or effects on the gene function may not be enriched even in the context of genetic drift. For example, it will be selected against if it's a tumor suppressor gene. So do you think that's a limitation of this approach?
So I guess what you're leading to is the idea of purifying selection of variants, rare variants that are damaging. And certainly, to some extent that occurs in these founder populations. But for the most part, these founder populations are relatively young. They're tens of generations. And through purifying selection, it's often the homozygotes that are not viable. So the heterozygotes still persist even if there's not selective advantage in some cases for those heterozygotes. So the bottom line is that yes, there will be some purifying selection, and of course those alleles we won't be able to identify particularly well, but I think there'll be many other cases in which that won't apply.
Is it
One
a
more?
quick question? Is
It's
it
a
a
quick
quick
one.
question?
So it really depends a bit on the filtering approaches that one uses, but certainly in many cases, particularly if you have a proband or just a trio, there can be multiple candidate genes that ultimately you can zero in on. But we like to sequence family exomes, so we try to get as many affected and unaffected family members as possible, and often that will allow us to whittle down to a single pathogenic variant. Okay, thanks.